Welcome to the DNA Lab's instructional video on DNA sample collection using a curiosity kit. The person whose DNA is being collected is called the donor. Use one sample collection kit per donor. To maintain confidentiality, no name should appear on any samples that you submit. There are a few things to remember before you begin. The donor should not eat or drink anything except water for 15 minutes prior to sample collection. Babies who are breastfeeding should have a sample collected as long as possible after their last feeding, a minimum of 1 hour, to ensure the DNA sample collected from their mouth is not contaminated with DNA from the breast milk. Avoid coughing, sneezing, and talking over sample. Do not open a second kit and start collecting a second sample until the first sample collection is fully completed and sealed. Before you begin, you will need a pen and an envelope labeled with the address of the DNA lab as well as your own address. The address of the DNA lab can be found on the instruction sheet provided with the kit. Each kit you receive will contain a foil pouch containing an FTA sample card and a pillow pack, a foam-tipped applicator otherwise known as a foam swab, and disposable gloves. The kit also contains the written version of these instructions with your personal file number written on the top right corner. Using new gloves, remove the FTA card from the foil envelope. Leave the pillow pack inside. Do not touch the pink part of the card. On the lines at the bottom of the FTA sample card, write your personal file number, the donor status, for example, the mother or alleged father, and the ethnic origin must be included for the alleged father's sample. For the alleged father's sample, please select one of the following ethnic origins: Caucasian, Black, East Indian, Oriental, Aboriginal, or Hispanic. Ethnic origin is only used for the purpose of statistical calculations. If you choose not to provide your ethnic origin, a default database will be used and indicated in your report. Use the foam swab to rub the inside of the donor's mouth for 20 to 30 seconds. Make sure to rub both sides of the foam swab firmly against each cheek, under the tongue, and behind the lips. If you are overly gentle, you will not get enough cheek cells on the foam swab. Open the FTA sample card. Do not rub the foam swab on the card. Simply press one side of the foam swab down in the circle of the pink section of the FTA sample card for 10 to 15 seconds. Turn the foam swab over and press the other side down for roughly 10 seconds. Part of the circle must change from pink to white to indicate that the sample has been transferred. It is crucial that you allow your sample to fully air dry before continuing on to the next step. Please remember that we require the donor's cheek cells, not their saliva, for DNA testing. If you have accidentally soaked the FTA card with saliva, you can place a clean tissue over the top of the card and leave it to dry overnight. Place the FTA sample card back inside the foil envelope. Note that the foil envelope now contains both the FTA sample card and the pillow pack. Remove the paper from the flap of the foil envelope and seal the sample inside. Label the outside of the foil pouch with the same information you labeled the FTA sample card with, which includes the file number, the donor status, and the ethnic origin for alleged fathers. Throw out the foam swab, the gloves, and any other disposables. Repeat all of these steps with a new kit to collect the next donor's DNA sample. Once all samples have been properly collected and labeled, place the foil pouches in a return envelope and mail it to our paternity office.